Welcome to this symposium on a global perspective of the narrow basis of intelligence. This is a topic that's been dealt with before in our OHBM meetings. And the reason for this specific symposium is the following. As has been logical due to the technological and scientific development, brain mapping was actually more intensively developed in some higher income countries and mostly in what we would call Western countries. In fact, the first large-scale brain projects that have provided big data that makes it possible to look into the details of what is the narrow basis of intelligence were at first limited to Europe, the US, and then gradually spread out. With the increasing availability and interest of brain mapping projects in different countries, we felt that it was important to bring a global perspective to this type of research because intelligence is a global phenomenon. It's a characteristic of human beings. Therefore, we have tried to bring together in this symposium diverse points of view regarding methodology, regarding area and country, economic development, and gender. And I think that this will help promote even more research in this area. We will have as presenters first, a general theoretical overview of the value of network science, which will serve as the backdrop for the rest of the presentations by Aron Barbey from the University of Urbana-Champaign. And this will set the stage for the rest of the presentations. We will then have one by Lippé from the University of Montreal, which not only will examine Canadian children, that is to say, children, but will look at a different imaging modality from other presentations that have been made in this Congress. It will be focused on electrophysiology, on EEG and evoked potentials, and it will look at the range of intelligence from those children that have intellectual disability to the other extreme of gifted children. We will then move on to China, that is to Asia, and from the Brain Nettone Center in Beijing, there will be a look at how there is gender difference in let's say, the narrow basis of intelligence based on functional connectivity and fMRI, and rooted in the work in the development of neuroinformatics tools from this institute. Finally, we will return to the Americas, to the Caribbean, and have a presentation by Maria Luisa Bringas, who currently works at the University of Electronic Science and Technology of China in the joint China-Cuba laboratory, about a project that was carried out in Cuba, which is the Cuban Human Brain Mapping Project, which gathered data on not only structural MRI and diffusion-weighted imaging, but also on EEG. But in this case, besides the new type of, let's say, this first example of a large-scale project from a lower middle-income country, and which is from the Caribbean. She will focus on the narrow basis of both fluid and crystallized intelligence using diffusion-weighted imaging. Now, the methodological work here, which is interesting, is the use of path models that are regularized for sparseness. So this will also add a new dimension in terms of methodology. So with this range of presentations, it only remains to say that we hope that this will continue to be a very interesting topic to be discussed also further in other countries, and that this will stimulate what we actually need, which is a global integration of work in general about human behavior and certainly about brain mapping. So we can now hear the next speaker.
Research in the psychological and brain sciences has long sought to understand the nature of human intelligence, examining the stunning breadth and diversity of intellectual abilities and the remarkable neural mechanisms from which they arise. Modern research in this effort is presented within our upcoming Cambridge Handbook, which reviews recent advances in the neuroscientific study of intelligence with an emphasis on theoretical innovation and emerging methods and technologies at the frontiers of contemporary science. The foundations of modern research were established in the early 20th century by Charles Spearman, who developed the correlation method and applied this technique to examine academic achievement. Spearman discovered that correlations in performance reflected characteristics of each discipline, noting that English and French agree with one another and having a higher correlation with classics and with mathematics. Beyond identifying the contribution of specific mental abilities, Spearman observed that the correlation among tests were always positive. This finding, which is now well established and called the positive manifold, provided evidence that all cognitive tests measure something in common. Spearman referred to this commonality as a general factor G, which represents the variance that's common across all tests of mental ability. These early findings motivated Spearman's two-factor model, which held that performance on tests of mental ability jointly reflect a specific factor S that is unique to each test and a general factor G that is common across all measures. Contemporary research has further elaborated Spearman's model to include an intermediate level of broad abilities that account for the variance that's shared across similar domains of cognitive ability. For example, the well-established Cattell-Horn-Carroll theory distinguishes between performance on tests of prior knowledge and experience, referred to as crystallized intelligence, from those that require adaptive reasoning in novel situations, or fluid intelligence. Taken together, the specific, broad, and general factors account for the hierarchical pattern of correlations that are observed among tests of mental ability. Spearman's discoveries ushered in a new era of research in psychology, motivating fundamental questions about the nature and origins of general intelligence. For more than a century, the cognitive foundations of G have remained largely invisible, beyond the reach of further scientific examination. This is because G is not a measure of specific knowledge, skills, or strategies for problem solving. These aspects of task performance are simply a vehicle for the measurement of intelligence. The general factor instead accounts for individual differences in information processing at a global level. Thus, we cannot understand the causal underpinnings of G by appealing to specific cognitive mechanisms or by directly examining the psychological tests from which the general factor is derived. Research on the nature of intelligence must therefore extend beyond psychology to examine the neural mechanisms that shape individual differences in cognitive ability. The interdisciplinary field of network neuroscience holds great promise for understanding the information processing architecture of intelligence, enabling the formal measurement and modeling of the interactions among network elements, providing a powerful lens for examining the emergence of global network phenomena. It was shortly after the discovery of G that Spearman's contemporary, Gottfried Thompson, proposed that the general factor represents a global network phenomena. Thompson held that G emerges from the interaction among the many elements of the brain, which he referred to as neural arcs or bonds. According to Thompson, each item on an achievement test samples a number of these bonds. He proposed that the degree of overlap among bonds accounted for the correlation between tests and the resulting positive manifold. Thus, Thompson's theory was the first to show that Spearman's discovery of the general factor of intelligence is consistent with a network perspective. Thompson's legacy can be found in modern theories, which posit that G originates from the mutual interactions among cognitive abilities. Individual differences in G are known to be influenced, for example, by language abilities, which facilitate a wealth of cognitive, social, and affective processes through mutual interactions. The central idea of the mutualism model is that change or growth in one aspect of mental ability is partially autonomous, owing to developmental maturation, and is also based on growth in other areas due to the mutual interaction between cognitive abilities. Research in network neuroscience provides a window into the modularity of cognitive abilities and their dynamic reorganization in the service of system-wide flexibility and adaptation. This work motivates the network neuroscience theory, which proposes that individual differences in general intelligence reflect network mechanisms for efficient and flexible information processing. The human brain is fundamentally designed for efficiency, to minimize the cost of information processing while maximizing the capacity for growth and adaptation. Minimization of cost is achieved by dividing the cortex into anatomically localized modules composed of densely interconnected regions or nodes. The spatial proximity of nodes within each module reduces the average length of axonal projections, increasing signal transmission speed and promoting local efficiency. Crucially, however, the deployment of modules for coordinated system-wide function requires a network architecture that also enables global information processing. Local efficiency is therefore complemented by global efficiency, which reflects the capacity to integrate information across the network as a whole. This complementary aim, however, creates a need for long-distance connections that incur a high wiring cost. Thus, an efficient design is achieved by introducing competing constraints on brain organization, demanding a decrease in the wiring cost for local specialization, and an opposing need to increase the connection distance to facilitate global system-wide function. These competing constraints are captured by formal models of network topology. Local efficiency is embodied by a regular network or lattice, in which each node is connected to an equal number of its nearest neighbors, supporting direct local communication. By contrast, global efficiency is represented by a random network, in which each node connects on average to any other node, including connections between physically distant regions. Recent findings suggest that the human brain balances these competing constraints by incorporating elements of a regular and random network to create a small world topology. A small world network embodies short distance connections that reduce the wiring cost, as well as long distance connections that provide direct topological links or shortcuts that promote global information processing. Together, these features enable high local and global efficiency at relatively low cost, providing a parsimonious architecture for human brain organization and capturing the modular and global network topology that is essential to general intelligence. Research in network neuroscience has consistently observed that the topology of human brain networks exemplifies a small world architecture, and this has been demonstrated across multiple neuroimaging modalities, including structural, functional, and diffusion tensor MRI. Alterations in the topology of a small world network have been linked to multiple disease states, stages of lifespan development, and pharmacological interventions, establishing the importance of a small world topology for understanding human health, aging, and disease. Evidence further suggests that intelligence is linked to characteristics of a small world topology. The discovery that global brain network efficiency is associated with general intelligence was established by Van den Heuvel and colleagues, a finding that was later replicated by multiple research groups. Further support for the role of global efficiency in general intelligence is provided by EEG studies, which demonstrate a positive association between intelligence and the small world topology of brain networks. To integrate evidence within this rapidly developing literature and to account for null findings, it will be important to examine how methodological differences among studies contribute to the observed results. For example, recent evidence suggests that functional brain network efficiency measured during cognitive tasks is able to predict intelligence more successfully than during resting state. A primary direction for future research is to examine how task-based network organization underlies general intelligence, moving beyond standard resting state paradigms. The widely recognized contributions of cognitive control to intelligence have motivated task-based fMRI studies, with an emphasis on mechanisms for adaptive problem solving and working memory. For example, fMRI studies show that working memory task performance is associated with an increase in network integration, particularly for frontoparietal and singular opercular networks. While significant strides have been made in identifying brain networks that underlie cognitive control, the contributions of these networks to specific facets of intelligence and the mechanisms by which they operate remain to be well established. An extensive literature in cognitive psychology investigates the cognitive and computational architecture of cognitive control and their contributions to individual differences in intelligence. An important next step will be to integrate cognitive theories with empirical data, investigating network mechanisms. Increasingly, scientists have proposed that cognition emerges from brain network dynamics, motivating an investigation of their role in general intelligence. Standard theories adopt a localizationist approach, identifying a fixed set of regions or networks that are central to general intelligence. Recent discoveries in network neuroscience motivate a new perspective about the role of global network dynamics in intelligence. Global information processing is enabled by the hierarchical community structure of the human brain, where modules are embedded within modules to form complex interconnected networks. Research in the field of network control theory further elucidates how brain network dynamics are shaped by the topology of strongly connected hubs, examining their capacity to act as drivers that move the system into specific network states. The hierarchical community structure of the brain may facilitate or constrain the transition from one network state to another. A direct path requires minimal transitions, whereas a winding path requires many transitions. Recent studies demonstrate that strongly connected hubs enable a network to function within many easy-to-reach states, engaging highly accessible representations of prior knowledge and experience that are a hallmark of crystallized intelligence. The capacity to engage easy-to-reach network states is exhibited by multiple brain networks, most prominently for the default mode network. This network is known to support semantic and episodic memory representations that are central to crystallized intelligence and to provide a baseline resting state from which these representations can be readily accessed. The introduction of weak ties is known to produce a more globally efficient small world topology. Weak connections enable the system to function within many difficult to reach states, reflecting a capacity to adapt to novel situations by engaging mechanisms for flexible, intelligent behavior. Unlike easily reachable network states underlying crystallized intelligence, difficult to reach states rely on connections and pathways that are not well established from prior experience, instead requiring the adaptive selection and assembly of new representations that introduce high cognitive demands. The capacity to access difficult to reach states is exhibited by multiple networks, most notably the frontoparietal and singular opercular networks. Together, these networks enable the top-down regulation and control of mental operations in response to environmental change and adaptive task goals. According to network neuroscience theory, the capacity of brain networks to transition between network states is supported by their small world topology, which enables each network to operate in a critical state that is close to a phase transition between a regular and random network. The transition towards a regular network configuration is associated with the engagement of specific cognitive abilities, whereas the transition towards a random network configuration is linked to the engagement of broad or general abilities. This framework predicts that early stages of learning depend on adaptive behavior and the engagement of difficult to reach network states, followed by the transfer of skills to easily reachable network states as knowledge and experience are acquired to guide problem solving. Development of fluid abilities from childhood to young adulthood is associated with individual differences in the flexible reconfiguration of brain networks for fluid intelligence. Recent evidence further shows that working memory training is associated with the engagement of cognitive control networks at early stages of learning, followed by increasing reliance upon the default mode network as knowledge and skills are acquired, consistent with the predictions of network neuroscience theory. Current research investigates how the flexible reconfiguration of brain networks is related to specific, broad, and general factors of intelligence, with particular emphasis on cognitive control. A central aim for future research is to clarify the precise role of brain network efficiency and flexibility in individual differences in general intelligence. According to network neuroscience theory, general intelligence depends on the dynamic reorganization of functional brain networks, modifying their topology and community structure in the service of system-wide flexibility and adaptation. Whereas crystallized intelligence engages easy-to-reach network states that access prior knowledge and experience, fluid intelligence instead recruits difficult-to-reach network states that support cognitive flexibility and adaptive problem solving. Thus, the capacity to flexibly transition between network states provides the foundation for individual differences in intelligence, supporting the rapid exchange of information across networks and capturing individual differences in cognitive processing at a global level. This approach stands in contrast to the view that individual differences in intelligence originate from a fixed set of regions or a primary brain network, and seeks to broaden the scope of analysis to incorporate evidence from network neuroscience on individual differences in the global topology and dynamics of the human brain. By investigating the foundations of general intelligence in global network dynamics, the rapidly developing field of network neuroscience will continue to advance our understanding of the cognitive and neural mechanisms from which individual differences in human intelligence emerge. Thank you.
Hi, I would like to thank Maria for organizing this great symposium. I'm sorry we cannot be all together today. This year is very different. I'm Sarah Lippé, neuropsychologist, full professor at University of Montreal, head deputy and scientist at CHU Sainte-Justine, and I direct the Neuroscience of Early Development Lab. What is intelligence? It's a very broad concept and very difficult to define. It's the ability to understand new ideas, to adapt to new environments, to learn from experience, to think abstractly, to plan, and to reason. It is an agglomeration of several cognitive functions and abilities, and they have to act in concert to translate into intelligent behaviors. And of course, the brain correlates of intelligent behaviors have to be multi-scale and multivariate. Why studying IQ and the brain correlates of IQ? Well, IQ correlates with success in life, social mobility, although less these days, job performance, health, and lifespan. Unfortunately, intelligence is quite different between individuals, from intellectual visibility on the left hand to giftedness on the right hand. What makes someone smart? How come we have children with high potentials and others with intellectual disability? Some of the researchers have thought that children with high potential could have a precocious mind. So this would mean that their brain would act like mature brains. They would process information more rapidly. They would habituate and use prediction more efficiently. They could deal with complexity better. And it would be the opposite for intellectual disability. Scientists have described brain maturation using electrophysiology for decades now. So when processing sensory information, brainwaves change with development, reduce in amplitude, reduce in latencies. We observe also more synchronization to stimulus each time a stimulus is presented with age. And there's also recruitment of higher frequencies also with age. The effect of repetition and the novelty effect are very basic brain processes. They allow you to sleep at night. And experimentally, with behavioral tasks, habituation in babies have been linked to cognitive function later in life. Electrophysiologically, typically when we see when we record the repetition effect, we see a decrease in brain response with repetitions. The novelty effect is an effect of surprise. So the brain typically increases in amplitude, and some component, specific ones like the P300, for example, are there to be analyzed. We studied the repetition effect, habituation, and novelty detection within a paradigm of the repetition of the vowel A and a local deviant I. With this paradigm, and this sequence being repeated several times, we can study repetition suppression, repetition enhancement, change detection, and learning of a sequence. We wanted to see in babies with a broad range of adaptive behavior how those brain response to these types of stimuli would act. As they are basic brain function, they should be measured very early on, and they should be able to discriminate between higher functioning infants and lower functioning infants. What we see in the brain response of babies when we repeat the vowel A, we see a decrement of brain response in power, in time, and frequency. And the amplitude resumes, the power resumes for the local deviant. We also see a decrease in synchronization of the signal, and which resumes for the deviant one. And we are interested in several frequency and time windows. We use the adaptive behavior assessment system as a proxy of IQ. In infants, it's difficult to measure neurodevelopmental quotient. Measures tend not to be so valid before 24 months because of the variability. But adaptive behavior assessment tend to be more precise and valid. What we see using a linear mixed model is that, yes, the signal decreases and shows the repetition suppression effect and resumes for the local deviant. And this model fit is increases with the GAC score, which is the Global Adaptive Behavior Score. Maybe even more interestingly, we see a repetition enhancement in the gamma range. And this enhancement is even more so as adaptive behavior increases. Also very interesting, we see a shift in shorter latency with repetition, and this shift in shorter latency is greater in infants that show better adaptive behavior. And in terms of change detection, we see that the comparison between the standard and the local deviant amplitude actually decreases with higher adaptive behavior, pointing maybe to the fact that infants with greater adaptive behavior have a better learning of the sequence. So we can see in babies with greater adaptive behavior that power in sensory response and habituation, together with gamma enhancement with repetition, increases with adaptive behavior. Repetition provides them with greater efficiency in processing sensory information, and they seemingly show greater learning of the sequence. And these data are thus compatible with the more precocious minds, although they will keep these precocious minds all their life and will be above other babies for all their lives too. So now we went on to see how gifted children would present differences in their brain signal. So we studied a cohort of children which is skewed towards the right in terms of IQ. We use a standard oddball task with standard deviants and also novel deviants, which were repeated 3 times each only. We saw an increase in phase synchronization with IQ in the theta and delta and theta bands. But surprisingly, we saw less synchronization for the deviant stimuli with IQ. And actually, this was confirmed with confirmed with the rare deviant, so the novel deviant that we repeated only a few times. With deviant change, so in the change detection, we saw more power in gamma. So although there was decrease with IQ of synchronization for change detection, we also saw an increase in gamma power. So it seems there's greater phase synchronization to the first standard stimuli, which would be an indication of a mature, healthy brain, but less synchronization for change detection. But this was accompanied with increase in gamma power. So maybe local processing of the stimuli within this frequency range for higher IQ kids. So there is an increase in synchronization, but it seemed to depend on the stimulus type. What happens in intellectual disability? Sensory processing and habituation have been studied for decades. Here we wanted to understand if all causes of intellectual disability would show the same brain signal signatures. So different syndromes actually affects different molecular pathways with different causes for each syndrome. We studied the Fragile X syndrome, tuberous sclerosis complex, SYNGAP1, which is a rare mutation, copy number variants, which we know affect IQ, and Down syndrome. In SYNGAP1, which affects the RAS pathway, cognitive development is greatly affected. We see IQ around 45 or 50. We compare to Down syndrome and neurotypicals. We see in the electrophysiological signal a decrease in latency and an increase in latency and decrease in amplitude. So less signal, less synchronization, less power. And this is confirmed by the phase locking value, where we see a delay and less synchronization in SYNGAP1. But this is not the case for Down syndrome. Using our repetition effect task with the repetition of a vowel in a deviant and a local deviant one, we compared several genetic mutations with very similar IQ levels. Tuberous sclerosis, SYNGAP1, Fragile X syndrome, and Down syndrome. And surprisingly, although we see a great repetition effect for the neurotypicals, we don't see repetition effect for SYNGAP1 and tuberous sclerosis, which was expected. But we see an habituation effect or a repetition effect in the Fragile X syndrome. We see this great amplitude, but also this decrease in the signal for the second presentation and third. This may be due to this task that is very simple and the fact that it captures a bit of attention because it's both visual and auditory. Because when we use a more complex task, like the repetition of pseudo-word, in neurotypicals, we see the greatest decrement in brain response with repetition, but in Fragile X syndrome, we don't see this decrement of brain response before the fourth presentation. And when we look closer at the data, we see that this decrement is mainly due to in children with Fragile X that have IQ higher than 50 otherwise. we don't see the repetition effect. Interestingly, when we look at the complexity of the signal, in this case it was with using spatial PCA, we see that in controls we need several components to explain the signal, or 60% of the signal. But in Fragile X, only 2 components are needed. So it really points to a very unitary and maybe too simple signal, at least in Fragile X syndrome, which may be a good marker of intellectual disability. So here, although we conceived IQ as a spectrum, principles that apply to high IQ individuals may not necessarily always apply to intellectual disability. And on top of this, intellectual disability syndromes, although showing similar IQ, do not always show the same brain response or properties. With this, I would like to thank my lab, especially for this presentation, my students, Gabriela Lopez in Gagnoth and Valérie Côté and Eve Lancette, and of course, collaborators and grant agencies. So with this, I will be pleased to answer questions and discuss further. Thank you.
Hello, everyone. This is Jing from Chinese Academy of Sciences. Today, my talk is about gender difference in connectome-based predictions of individualized intelligence content and subdomain scores. So we know that individual differences in general cognitive ability can be measured by the IQ scores, which assesses ability including planning, reasoning, comprehension, abstraction, and learning. IQ scores are strongly predictive of various life outcomes, including educational achievement, occupational attainment, social mobility, and job performance. And since intelligence is postulated to draw on communications between regions that are responsible for high-order cognitive functions, there are many studies have been carried on to studying the associations between IQ and brain imaging. And we could find here are all those studies which has been carried using the gray matter, white matter, cortical thickness, and function connectivity. Well, among all those findings, the prevailing comprehensive synthesis on intelligence is the parietal frontal integration theory, that is for short, PFIT. Recent work has suggested that intelligence is underpinned by the communication between widespread brain regions, including but not limited to the PFIT areas. However, the gender difference on IQ and its subdomain prediction using brain imaging measures has not been fully studied. While on the other hand, the field of brain science and medicine is moving forward to use of individualized biomarkers, and the neuroimaging techniques provide the opportunity to estimate individualized cognitive ability with objective and tangible brain-based measures. There are many wonderful reports, and here I just list a few. So therefore, in this study, we have 4 aims. So we want to check whether the individual IQ scores could be predicted using the refined whole brain function connectivity. And the second is we want to see whether there is a gender difference between IQ prediction using FCP. And the third is we want to assess the gender specificity of those male-specific and female-specific IQ predictive FC features. And the fourth is if I want to evaluate the generalizability of those IQ predictive models using a different independent data set. So here we have 3 data sets. The first discovery data set including 360 healthy young college students, almost half female and females. And there are 2 validation data sets. The first is HCPQ3, which including 200 healthy adults. And here in this data set, the fluid intelligence was used instead of those WAIS IQ. And the second validation dataset is Cobra, which including 120 subjects, which include not only health controls, but also schizophrenia and bipolar disorder. So we want to test whether the IQ predictive models could be generalized to those intelligence equivalent measures and also both controls and patients. And here are the flow chart. We could see that we first using a Brinatium atlas to extract the refined FC, which has 242 FCs, and then we used several feature selection methods to doing those feature elimination and then using those different regression models to build that. So here we can see that we used the one out with tenfold cross-nested validation. And in those discovery dataset, we want to identify those most selective FCs. And both those FCs, we build models to test in validation dataset. And here you could see that we just test 4 directions: IQ specificity, IQ predictability, gender specificity, and generalizability. So first, in the models, we have tried 8 models. Among these, Relief F plus Lasso achieved the best performance. And the highest for female is 0.72, and the highest correlation between estimated IQ scores with true scores for males is 0.46. And we want to see if this is really a gender difference. We divided the whole samples into 2 halves and reran the prediction procedure with each subset for 100 times. The results shown that the correlation difference between randomly divided subsets was much lower than that between males and females. That is the green bars. So the difference in predictability was a real reflection of the actual gender difference, which is not divided by the data division. And also in each run for the female and males, we identified those most selected ICs in every iteration. So there are 13 and 8 consensus FCs were derived for female and male respectively. So in the later presentation, we call those most contributed FCs like a consensus FCs. And then we first check whether these FCs has a specificity to intelligence. And when we correlate these FC patterns with IQ main score and subdomain scores, and also those personality scores, which is intelligence irrelevant, we could find that the FC patterns were uniquely correlated with IQ and its subdomain score only in the same gender, that is in those boxes, but neither for the opposite gender here and not for those IQ irrelevant measures. So which shows that the identified FCs were only specific for intelligence. And then we use these FCs to build models for the prediction. And here we could see that the red bar is using male FCs to predict males. The blue is for male predict male, and the red is female for female. And the light blue and the light red is for those opposite gender prediction. And you could see that in all those significant predictions, females is higher than males, and only within the same gender, the prediction is significant. The prediction cannot be done across genders and also in those IQ irrelevant measures such as temperament. And finally, we used these FCs to build the linear regression model. And based on the same model, same feature, we expanded to the HCP data and Cobra data. And we find that robust predictions of IQ capabilities were achieved across 3 datasets, which is in total 680 subjects and 2 intelligence measures, that is IQ and fluid intelligence. And when we look closer, which FC nodes really works, and here there are the weights distribution. And we find that both males and females have nodes lying in those fundamental P-fate indicated regions, such as parental and inferior frontal. While for the males, a more distributed network, including lingual gyrus and some cortical areas, were also identified. And also there are some female-specific regions, including fusiform, and male-specific regions, including basal ganglia and thalamus, were identified. So to summarize, in this study, we find that IQ can be predicted by using patterns of refined FC, and a relatively high resolution pixelation contribute to the detection of individualized variability. Also, for the predictions estimated using those FCs, only the same genders works. And also the females have more predictability than males. Also, we identified the prevailing P-fate implicated regions were common among both males and females, which underpins its crucial role in the biological basis of intelligence. While for the females, the predictive FCs were mainly concentrated on those fundamental regions, while males have a more wider subcortical regions, including amygdala, basal ganglia, and the thalamus. And finally, we find that there is a significant gender specificity on these FC features that are really predictive for the IQs. So this work has been published in the cerebral cortex. And our group also did some further work. For example, we also predict IQ using multimodal data that is based on the cortical thickness and the refined FCs. And in this study, we find that the male IQ demonstrated higher correlation with regions involved in spatial cognition and logical thinking, while females, the more IQ related is mobile learning and item memory. This is consistent with their respective superiority in the cognitive and behavioral performance. And also our lab did some individualized prediction work using neural markers. We have provided an overview that utilize machine learning approaches to identify neuroimaging predictors in the context of cognitive function, symptom severity, and personality traits. So that's all. Here is my group. I would like to thank all my collaborators and lab members. And here is our posters for this OHBM. Thank you all. Any questions?
Okay. Thank you for this invitation for the first virtual human brain mapping meeting. My presentation is about crystallized and fluid intelligence, how they can predict the microstructure of a specific white matter tracts. The white matter and intelligence has been a very hot topic for many years. The main tendency in this research is the aging and neurodevelopment. But the research are mostly concentrated on fluid intelligence or G factor. And one problem we have in this research is the lack of a standardization of the instruments to assess the IQ, specifically in the big global brain initiative. Also, the human brain mapping already addressed this topic before in Vancouver meeting. And we have the motivation to replicate the watershed model of Roger Kiewit in our data set. The main highlights of our research are, first, we have the inspiration in the watershed model, but we employ the multiple indicator, multiple causes model, MIMI, with regularization. Second, we use another cohort from a non-Western country with low income. And besides, we use the white matter tracts based average, fractional, anisotropy, but estimated in individual native space. And adding more IQ variables, the 4 indexes of the Wechsler Adult Intelligence Scales. Why is it important to use another cohort from a non-Western low-income country in Latin America? Because most of the research in intelligence are using highly educated Western countries in very high-income countries. But where is Cuba? Why is Cuba interesting? You can see in the map the red, a small island in Central America. It's lower middle income economy, highly educated, with very blended population, with ancestry for Africa, Spanish, Spain, Lebanon, Chinese, low children mortality, and the health system is universal and free. Also, we have an integrated research environment. One of the achievements is the fast and controlled COVID epidemic. Other achievement we have with this environment is the Cuban Human Brain Mapping Project. It's multimodal because has the clinical variables, electrophysiology, MRI, neuropsychology, and also the sample is the healthy right-handed participant, 83 healthy right-handed participant, age 35 years old, and education 12 degree. The instrument we employed was the David Wechsler Intelligence Scale for Adults. To the left, you can see the structure of the test. The verbal IQ and performance IQ and 4 cognitive domains. Verbal comprehension, working memory, perceptual organization, and processing speed. From this structure, we try to test 2 latent variables, crystallizing intelligence related with the verbal IQ and fluid intelligence related with the performance IQ. To the right, you can see the performance of our sample in the main scales of this test. On the other hand, we need to create our own non-Caucasian statistical track-based average atlas. You can see on the to the left, we estimate, using the Mori space atlas, the ROI, the region of interest. Later, we use the same regions of interest in the native space of the subject. Using this software, the fiber assignment for continuous tracking, we create our atlas. For the test of the hypothesis, how many latent variables we need for the research, we use confirmatory factor analysis. We can use first the general intelligence latent variable, or we can use 2 latent variables based on the Bayesian information criteria. And this second model for the 2 latent variables was more robust. Later, after that, we employ the multiple indicators, multiple causes, the mimic model, to know the relationship between the neuroanatomy and behavior. And we need to solve the problem of the high dimensionality of the IQ and information from the white matter track basis measurement. We use the regularization plus the mimic to select what the relevant variables for this study. Using this method, this is the result we obtained. First, we find novel association of the fractional anisotropy with the four cognitive domains, mediated by two latent variables, the crystallized and the fluid intelligence. You can see in red the positive and the green the positive and the red the negative relationship between the latent variables and the principal tracks and also the age. In summary, exploring the full set of the indices of the Wechsler scale, it was possible to find novel association of the fractional anisotropy with the four cognitive domain mediated by the latent variables. The fluid intelligence was related with the superior longitudinal fasciculus and crystallized intelligence with the forces minor. This relation with the forces minor tract with crystallized intelligence was only possible to discover here because we included in the model more indices related with this domain. Something that seems to have been omitted in many studies before. What we need for the future? Okay, we need more international collaboration, more diversity in ethnic, education, age, and socioeconomic status, joint protocols to unify the tools, the analysis of the approach, and we need to add many other variables, multiomic, electrophysiology, and Bayesian modeling, for example. This data is available. You can see the data at the Human Brain Mapping Project, the Cuban Human Brain Mapping Project at Cuban Neuroscience Center, and very soon it's going to be public in LORIS. LORIS is the Longitudinal Online Research and Imaging System in McGill University in Canada. Also, the data was published and the research in the Human Brain Mapping journal. Finally, we need to recognize the contribution of many people in the joint China-Cuba lab for neurotechnology for the Cuban Neuroscience Center. And especially, we need to recognize the contribution of Dailin Góngora. This research was part of her PhD dissertation. Thank you very much for your attention.